So I'm recording this video because I found a really cool tool. I mean, I knew about Allen Brain Atlas and I've been looking into it a little bit, but for those of you who are working on developing your independent projects, either whether you're in Neuro 250 right now, or if you're in Neuro 350 and you're just interested in mental health disorders, or if you're working on thinking about your independent project that you're going to create and develop in, and further work on in Neuro 399 and then doing for your Neuro 498 course. Because of the pandemic, we have to be remote, and so it's more difficult to do the research on campus, and we don't know when that's going to change. And so in order to create a plan to get your work done, I wanted to show you this tool. Or even if you've already gone past that, this is just really interesting to look into. So at Allen Brain Atlas, and I will post this link here, but this is their human brain tool. So I went to, and I selected human brain. You can look at mouse brain, developing mouse brain. You can even go to the developing human if you're more interested in that. So within the atlas, going to the human brain, and then clicking on there, you can do microarray data. It's difficult to process, but if you just spend some time with it, you could get it done. They also have in situ hybridization data, and you can also go to MRI. But the one that I thought was interesting, because they have there's a lot of information packed within it, is this in situ hybridization data. And then you just do a gene search. You can look at all these different studies too, but if you have a particular gene that you know of, so I know, let's see, someone's looking at the renin gene, so you can look up renin, and that one dropped down some different receptors. So you're looking at all the data, so that narrowed it down, so they didn't have as much there. But if you leave this clear, and then you can go and select a gene category. So anything that's involved in Alzheimer's disease, you can click on that one. Or autism, or depression. So any genes that are related to those are going to pop up in this window down here. The one that we're focusing on in Neuro 350 is schizophrenia. So I'm going to select that one. And then, so it pops up, so that ASD, so the schizophrenia related ones pop up in the autism spectrum disorder study. But then there's a specific schizophrenia study where you have a male and 2 females and 2 males, right? And they're looking at the same region of the cortex. If you want to look at a specific brain region, so let's say we wanted to get the hippocampus included in there, then we can click on this and see if anything pops up. Okay, there's only one that pops up in there, so that might not be the greatest one, but there are likely more that are popping up in different areas of the cortex. These ones happen to all be control, so if we are looking at specific ones, it looks like the ones that are on here are just kind of going to show me controls there. But I think the ones where they were looking more in the cortex, there were some, so you can look by different brain regions, or you can just go, let's go back and clear my specific regions. How do I do that? There we go. So I cleared my specific sections, and now you can look at different individuals who were diagnosed with specific things, either their controls, or you can find ones that are specific to schizophrenia if you wanted specific regions, or if you wanted to only look at males within a certain age. But let's check this person out. If we click on that, right, it tells you the different genes that they looked at within that specimen. And then you can go over here to open in situ hybridization data. And then it's usually only one section of the brain, but what you'll see is a in situ hybridization of a specific protein. and then a reference image over here. So this is just like a Nissl stain, where it's just staining all the cell bodies. And you can navigate through that particular section. So there's data analysis that you could do here, right? You could count the number of cells, which would be that are positive for that particular gene, and compare it to a control within the same area. If you go to, so there's a bunch of different, looks like there are 43 different ones here. So now you look at this one, and this one looks different. There's not as much positive staining. So it was a different, so this is the, I think, different receptor from the last one. But the idea is that there's a whole bunch of data here looking at specific things that you could actually measure and ask your independent question on, so that you can do your research without having to be on campus. So I recommend playing around with it. It's interesting no matter what, right? Just to be able to look at these. But if you have a question, like if you want to know about the gene expression of a particular protein, or sorry, the in situ hybridization, right? You're looking at the RNA, but then you can, for the most part, extrapolate the protein expression from the RNA expression. But it's just really cool and a really good tool. Look at the neurons! I love it. Anyway, so if this would be a good tool for your research, I recommend going in and just spending some time in these images, and then picking something that might interest you, and then seeing if you can develop a research project around it. And always let me know if you have questions. And the goal for all of this, right, is to get you asking those independent questions. And now we have the tools available, since the Allen Institute has done all of the lab work for you, right? And all the data is there. They're just waiting for people to analyze it in a unique way. So that's your job, is to analyze things in a unique way with the data that you already have.